Welcome to our explorations of recombinant DNA. In this video, we'll break down what recombinant DNA is, the step-by-step processes behind it, its importance in science and medicine, and a real-world example that changed the course of healthcare. So, what is recombinant DNA? Simply put, recombinant DNA refers to the DNA that has been artificially created by combining genetic material from different sources. Scientists take specific genes from one organism and insert them into another organism's DNA, creating something entirely new. This groundbreaking technology allows us to manipulate genes in ways that were previously impossible. It's used to produce life-saving medicines, genetically modified organisms, and even to explore new treatments for genetic disease.
Now, let's walk through the process of creating recombinant DNA step by step. The first step is gene isolation. Scientists start by identifying the gene they want to know with. This could be a gene for insulin production, for example, or a gene that produces a helpful protein. To extract the gene, they use special tools called restriction enzymes, which act like molecular scissors, cutting DNA at precise location.
Next, the isolated gene needs a vehicle to carry it into the new organism. This is where vectors come in. Vectors are like delivery truck for DNA. The most commonly used vector is plasmid, a small circular piece of DNA often found in bacteria. Scientists insert the isolated gene into the plasmid to create recombinant DNA.
Once the gene is inside the plasmid, the next step is gene insertion. The gene is inserted into the plasmid using an enzyme called DNA ligase, which seal the gene into place, making sure it's part of the plasmid's DNA.
Now, we have a recombinant DNA. The next step is called transformation. This is when the recombinant plasmid is introduced into a host organism, such as bacteria. Through transformation, the bacteria absorb the new DNA and start using it to make whatever the new gene codes for.
At this point, the host organism begins expressing the new gene, meaning it starts producing the protein or substance that inserted gene codes for. In the case of bacteria, they might start producing human insulin or even enzymes to break down waste materials. Let's look at a famous example, the production of human insulin. Before recombinant DNA technology, insulin had to be extracted from animals, but now, thanks to recombinant DNA, we can insert the human insulin gene into bacteria, allowing the bacteria to produce insulin on a large scale. This insulin is harvested and used to treat diabetes worldwide.
Recombinant DNA technology is used in many important applications beyond insulin. It's the foundation of gene therapy, where genes are inserted to treat genetic diseases. Genetically modified organisms or GMOs, like crops that are resistant to pests, and even vaccines where recombinant DNA is used to produce safe and effective vaccines.
While recombinant DNA has incredible potential, it also raises important ethical questions. How should we regulate gene editing? What are the long-term effects of genetically modified organisms in the environment? These are questions scientists and ethicists continue to explore as the technology advances.
To recap, recombinant DNA technology involves isolating a gene, inserting it into a vector, transforming a host organism, and allowing it to express the new gene. It has transformed medicine, agriculture, and biotechnology, unlocking possibilities that were only dreamed of. Recombinant DNA is more than just a scientific breakthrough. It's a powerful tool that's reshaping the future. Thanks for watching, and remember to stay curious. Don't forget to like and share and subscribe for more amazing science videos.